Okay, in this video I'm going to go through more details on respiration. So we're talking about aerobic respiration here, taking oxygen and using it to break down the sugars as much as we can. We've already done glycolysis, which can happen in aerobic or anaerobic, and now we're going to get into more of the details after glycolysis, what happens if it's a cell that does have a mitochondria. So the first step is called the preparatory conversion. Sometimes you'll see it called the transition stage. This is necessary to begin the Krebs cycle because the Krebs cycle uses a substrate called acetyl-CoA, and we have pyruvate. So we have to change pyruvate into acetyl-CoA to start the Krebs cycle. This happens in the mitochondrial matrix, and let me just show you the structure of a mitochondria. So a mitochondria like this. In the past, I know that you have always drawn this little squiggle. And so this time I'm just going to show you that that squiggle really represents an inner space. The inner space is called the matrix, M-A-T-R-I-X. So the matrix is in here. This is what we call the outer compartment. Okay, so this outer space here, outer compartment. This is the outer membrane, or outer mitochondrial membrane. And this here is the cristae. The cristae is the inner mitochondrial membrane. So you'll remember that a chloroplast has a double outer membrane. The mitochondria also has a double outer membrane, but one is much further in. This cristae, the lining that's holding the matrix in, is the second membrane, the inner membrane. So this preparatory conversion that we're talking about happens in the matrix, so it happens in this middle space. In the preparatory conversion, one carbon gets removed from pyruvate. And you remember that pyruvate was a 3-carbon molecule. So we're going to break off one of those carbons. That gives us carbon dioxide, and it gives us acetyl-CoA. The enzyme is called Coenzyme A, and it does this job of removing one carbon off the end, producing the beginnings of carbon dioxide. Most people can remember respiration like this. Here I am respiring. I'm taking in oxygen and I'm breathing out water vapor, so water is going to be one of our products, and carbon dioxide gas. So carbon dioxide gas, the first production of it happens here in the preparatory conversion. The 2-carbon molecule called acetyl-CoA can now go into the next step, which is the Krebs cycle. This also did something really important. NAD+ is a coenzyme that is going to be needed for the last step. It's going to be needed for oxidative phosphorylation. And if you remember in glycolysis, we also built 2 NADH molecules. So these NADH molecules that we're stockpiling are going to be really important for not substrate level phosphorylation, but instead utilizing that electron transport chain and doing chemiosmosis and having a greater output of ATP production. All right, Krebs cycle. This is our next step. This is our big step that's happening within the mitochondria. It's happening in the matrix, and this cycle is how we get the majority of our carbon dioxide. It begins with acetyl-CoA. It fully breaks it into 2 carbon dioxides. Those are what get released. So we have an abundance of carbon dioxide. It gets released out of our body because we are producing it every time we break apart sugar. It reduces 3 NAD+ to 3 NADHs. So it is reducing that. We also have a new coenzyme here. It's called FAD. Oh, I shouldn't even have an H on there. Just FAD to FADH2. So this is another coenzyme, another molecule that again we're stockpiling for the end. It produces 1 ATP, so that's just substrate level, meaning the enzymes are doing the production. And then I also want you to remember all of this is really multiplied by 2 because one glucose molecule gave us 2 pyruvate. And the 2 pyruvate each formed acetyl-CoA, and each acetyl-CoA goes through these steps. In class, I'm going to do a big diagram with you, and we'll talk about the numbers, and we'll make sure that all the math makes sense. All right, the last step of respiration. This is where we're really going to get the energy because as you've seen, we only made 2 ATP in glycolysis, none in the preparatory conversion, and 2 in the Krebs cycle. That's lame. That is not enough ATP for our bodies to run. We need a lot of energy. So we get that by oxidative phosphorylation. This name really gives us a lot of information. Oxidative means there's going to be things that are losing electrons. It also sort of reminds us this is the step where we need the oxygen. Phosphorylation, you already know that word. That means to add a phosphate group onto ADP. Our goal is to phosphorylate and make as much ATP as possible. This occurs in the inner membrane. And you'll remember we called that membrane the cristae, C-R-I-S-T-A-E. Coenzymes, we are now going to put these to work. NADH and FADH2 are going to be oxidized. They're going to lose their electrons, and they lose it to an electron transport chain in the membrane called the cristae, the inner mitochondrial membrane. NADH loses its electrons very early in the chain. FADH loses theirs a little bit further down the line. As the electrons move through the chain, just like in photosynthesis, we're going to pump some H+. The movement of electrons through the chain allows H+ to be pumped into the outer compartment. Now, here's the difference. In photosynthesis, we were pumping them into the lumen, into the inner space of the thylakoid. Now, they're actually getting pumped to the outer compartment. So that is one difference that you need to remember from photosynthesis to respiration. The H pluses are in the outer compartment. That builds up my proton gradient, and now the electrons are pulled through the chain by oxygen. At the end of the electron transport chain, there's oxygen. Very electronegative, very able to attract electrons to it, pulling those electrons. Which is another reason why you have to have oxygen to do this aerobic step. At the end of the chain, oxygen gets reduced, so it gains the electrons and it forms water. So we formed carbon dioxide in the Krebs cycle. Here's the water part. Then, for every NADH that gets oxidized, 3 ATP are produced. And how are they produced? I hope that you are remembering this, ATP synthase. ATP synthase is that special channel protein allowing the hydrogen ions to go with their gradient from the outer compartment into the matrix. So as they move in, the rotor spins, ADP and phosphate come together producing ATP. For every NADH, 3 ATP are made. For every FADH2, 2 ATP are made. Whoops, I don't want that. Alright, so let me go down to here. So, for every FADH2 oxidized, 2 ATP are produced. Alright, I wanted to do a little bit of math at the end so you have it in your notes. Again, I'm going to do it on our big concept maps, but just to hopefully help it make sense. Final ATP counts, this is all per glucose. So, glycolysis, I made 2 ATP, and then in green I put here 2 NADHs are made. I'm going to want a count of how many NADHs and FADH2s I've produced, because those are going to be like coins that I turn in for ATP at the end. The next step, the preparatory conversion, 2 NADHs are made, but no ATP. The Krebs cycle, 2 ATP, 6 NADHs, and 2 FADH2s. Those numbers all might seem boosted up. That's because I realized when I have 1 glucose going in, I had 2 pyruvates, therefore 2 acetyl-CoAs. So that's why the numbers are all doubled. I want this to be how many am I making per glucose. All right, my last step, my oxidative phosphorylation. This is really where I get the good bang for the buck. Here it is. 10 NADH are oxidized. Here they are. Whoops. 2, 4, 10. That is where I get my numbers from. 3 ATP each, so that's a total of 30 ATP. Oxidative phosphorylation is giving me much more ATP. This is why it's so important that large organisms like us are able to do aerobic respiration, getting more energy out of 1 sugar. Then I had 2 FADH2s. Both came from the Krebs cycle right here. Those 2 each gives me 4 ATP. So that is 34, 35, 36, 37, 38 ATP that I could make per glucose. So there's a little note that I put down here. I call it shipping costs. To bring pyruvate, a pretty large 3-carbon molecule, into the mitochondria takes some energy. So to move that in, because sometimes it's going against its gradient, plus it's large in size, that usually takes 1 ATP each. So that brings us down from 38 ATP per glucose to 36 ATP per glucose. And anaerobic only did glycolysis, so it only produced the 2 ATP in comparison. You will see this is a drastic difference, and that depending on the organism, some organisms might only get 34 ATP per glucose. Sort of depends on how their enzymes work and what energy they expend in the process. But obviously an organism that is eukaryotic, has mitochondria, takes in oxygen, wants to utilize the sugar bonds as much as possible, producing as many ATP as you can. Go through your textbook. It has some great diagrams. I want you to look